Okay, so when we talk about these eukaryotic chromosomes are inherited in sets, and you talk about homologous chromosomes as being very similar, there's usually less than 1% difference between those homologs. Remember, one's from mom, one's from dad. So that means that for every 100 nucleotides, one of them is different if you look at the sequence. So nevertheless, the slight differences that occur, which is not a whole lot, can lead to allelic differences. So for example, if we look at eye color, which is associated with this gene called OCA2, which is part of the system that's involved in converting tyrosine into L-topa. That's part of those particular enzymes, and doing that to produce melanin for the eye. So blue allele versus the brown allele. For the brown allele, it produces a protein called the P protein. We don't necessarily know the understanding of the function of this protein, but it brings about brown colored eyes. Whereas if you have a mutation in that OCA2 where it cannot produce the P protein, then what's left is eyes that are blue-eyed, which have no pigmentation in those eyes. So that gives that the distinctive blue color that you see. Melanin is an interesting structure. Melanin is basically created by cells called melanocytes, and they go through this process to produce what they call a melanosome. So there's little membrane-bound structures that end up soaking up all the melanin and producing the melanin in them. And those particular little packets of melanin, called melanosomes, are then given to various cells. They're distributed to cells so that you have this pigmentation occurring. So for example, in our skin color, each one of the cells of our skin receives these packets, a certain amount of these particular packets. Now the skin will break them down at different rates, thus giving us a variation in skin tone or eye color, different eye color tones. So that's what happens with that particular structure. In this particular one, OCA2 is very much involved in the production of the P protein to bring about a brown colored eye. Now the eukaryotic organisms are inherited as sets, okay? So the sex chromosomes are not homologous, all right? They're not homologous, especially X and Y. They're not homologous. Now for a woman, yes, hers are going to be homologous. They're going to have the genes in the same location. But for a male, you only have one X chromosome, you only have one Y chromosome. So whatever the genes are here, they're going to be expressed versus the Y chromosome. So they differ in size and genetic material, as you can see in this particular image here. However, they do have a short region of homology. It's called the pseudo-autosomal region. It's up here called PAR1 and PAR2. So these guys here, they are where pairing can occur during meiosis to make sure that they pair up properly so that they can be separated. There's about 1,400 genes associated with the X chromosome. The vast majority of them are not related to sex determination. However, in the Y chromosome, there are about 83 genes for sex determination and sperm production. So that's where those particular ones come from. You don't find those over here in this particular one. Okay? So these PAR regions allow for pairing during mitosis or through meiosis to occur. Now, homologous chromosomes have the same loci. Okay, so here the loci is going to be the physical location of a gene on the chromosome. It's called a locus. Okay, so locus is going to be the non-plural format of this, or singular format of this. So A and A, this is the location for the chromosome A. B, notice here, this is one allele of this particular gene. This is the other allele of this gene, okay? So this would be a heterozygote. Here's a homozygous over here, homozygous at this point. So this is homozygous for the dominant allele. This is heterozygous for this one. And this is homozygous for a recessive allele. So each one of these is a gene loci. Okay, this is a gene loci that are associated. All right. Now, cell division. So we've talked about chromosomes, now we have to talk about cell division, which is really basically set up to be able to divide the chromosomes and the cytoplasm evenly if you just do a simple, straightforward cell division, which is mitosis. So one purpose of cell division is called asexual reproduction. It's not involved in fusing any types of gametes together. A new individual develops from either a single cell or from a group of cells. So yeast, multicellular fungi, cuttings of roses, and fruit flies. So that can be called asexual reproduction. The second important form is cell division is multicellularity. Plants, animals, and fungi are derived from a single cell that has undergone repeated cell division. So humans start off as a single fertilized egg and end up being an adult with 30 to 40 million, possibly 100 trillion types of cells, 100 trillion cells. So this is sort of a conservative number, and some people are a little more liberal with the number, and they say it might be 100 trillion. So sexual reproduction can occur at this level for multicellular, the fusion of two haploid gametes to produce a single diploid zygote cell. There's an alteration between haploid and diploid phases, two cell types. We have a somatic body cells, which are 2N, and then we have the germ cells. And these have actually, the germ cells literally separated themselves during our embryonic development. So as our embryo was developing into us, the fetus, those germ cells actually left the body of the embryo and set outside because there's such a very strong hormonal growth factor environment that would have influenced their particular structure. They were supposed to become germ cells. After that's done, then they divide and start crawling back into the embryonic body, and they head for what they call the urogenital ridges. And in those ridges, part of those ridges are going to become the gonads, okay? And they enter into the gonads, continue to divide, and they form whether it's going to be a testes or an ovary, depending on the sex of the individual. And so they then have not been exposed to the environment of the developing individual, and they retain that germ cell status. So prokaryotes reproduce in an asexual manner, and we refer to it as binary fission. The capacity of bacteria to divide is really quite astounding. They can divide in like 20 minutes, very rapidly, okay? I look at eukaryotic cells, usually it's somewhere in the process of around 18 hours or longer for their particular division. Prior to division, they replicate their DNA. So here the DNA is replicated, and then they divide into two daughter cells. They divide and evenly separate the amounts of DNA and the cytoplasm to create the daughter cells. It does not involve a genetic contribution from two different gametes in this situation. Eukaryotic cells, they will actually go through a cell cycle. So they have to go through various stages of development. They go through what we call G1, that's part of the cell cycle, or the growth phase 1. Then they go through a DNA synthesis, okay, so they double the amount of DNA during S phase. And then they hit G2, which is growth level 2, growth stage 2. And in this stage, they actually have double the amount of DNA, and it's all about preparing for the M phase, which is going to be where mitosis occurs. So you go G1, S, G2, and then you go through the stages of mitosis: prophase, prometaphase, metaphase, anaphase, telophase, and then cytokinesis. So we're going to go over these in association with mitosis. So, mitosis and cytokinesis. Basically, you're going to go through these gaps or growth phases, G1, G2, S phase, you have M phase here. We'll talk about these. Some cells may remain for long periods of time in what we call GO phase, where they separate out to this, and they might have a permanent withdrawal, and they'll end up basically go into a differentiated cell type. Some, on the other hand, will actually enter into this GO phase and become transient withdrawal. They'll just sit out here like stem cells. These would be like stem cells. Whereas some have actually made a decision, and they're in the process of differentiating into various cell types. So we have it located in different regions of our organs. We have stem cell niches, and these particular areas are designed to do repair for us. We have niches even in our brain tissue, and when there's a damage done, these things will start to produce, based upon the signals, they'll produce new different cells that are on the pathway differentiated. These cells will actually literally crawl to that region to help do the repair. So it's very important for us to understand how this works, so we can even understand how stem cells work.